What's the best method for RNA extraction? It's a question that's probably crossed your mind more than once. And it's a valid one, as the quality of RNA extraction is crucial to the success of many research procedures. RNA, or ribonucleic acid, is one of the three major biological macromolecules that are essential for all known forms of life. Its extraction is a key step for numerous laboratory procedures, including gene expression analysis, genetic engineering, and molecular cloning. In our quest to find the best method, we'll be exploring two main contenders, the spin column method and the tryzol method. Each of these methods has its strengths and weaknesses, and your choice may depend on your specific needs and the resources at your disposal. But don't worry, we'll guide you through the intricate details of both methods. Let's dive into these two methods and see which one might be the best for your next PCR, cDNA, or RT-PCR. First up, we have the spin column method. This technique is a popular choice for extracting RNA, and for good reasons. Let's dive into the step-by-step process. The spin column method starts with the disruption and homogenization of the sample. This is typically done mechanically, using a pestle or a bead beater to break down the cell walls and expose the RNA within. The sample is then treated with a lysis buffer, which disrupts the cell membranes and releases the RNA. Next, the sample is loaded onto a spin column. This is where the magic happens. The column contains a silica membrane that binds the RNA when centrifuged. The unwanted cellular debris is then washed away, leaving only the RNA bound to the column. The RNA is then eluted from the column using a low ionic strength solution or water. This solution disrupts the interaction between the RNA and the silica, allowing the RNA to be collected in a separate tube. Now let's talk about the advantages of this method. The spin column method is known for its speed. You can go from sample to purified RNA in less than an hour. Additionally, it produces high-quality RNA with minimal contamination, making it an excellent choice for downstream applications, such as PCR and RT-PCR. However, no method is perfect. The spin column method does have some downsides. For one, it requires specific equipment like a centrifuge and the spin columns themselves, which can be costly. Also, while the RNA yield is usually sufficient for most applications, it's generally lower compared to other methods like the tryzol method. Another thing to consider is that this method doesn't allow us to skip DNA extraction. So, if you're looking to get rid of any potential DNA contamination, you'll need to treat your RNA sample with DNAs. That's the spin column method in a nutshell. Now let's look at the tryzol method. Now let's turn our attention to the tryzol method. The tryzol method is a well-established procedure for extracting RNA. It's a bit like an all-you-can-eat buffet, but for nucleic acids. It extracts not only RNA, but also DNA and proteins, which can be a major advantage. The process starts with cell lysis, where we disrupt the cell membranes to release the nucleic acids. This is achieved by adding trizol reagent, a solution that contains phenol and guanidine isothiocyanate. The phenol denatures proteins, and guanidine isothiocyanate helps to inactivate RNases, enzymes that would otherwise degrade our precious RNA. Next comes phase separation. Chloroform is added and the mixture is vigorously shaken. This step separates the solution into two phases. The upper aqueous phase contains RNA, while DNA and proteins are in the interphase and organic phase below. The RNA is then precipitated from the aqueous phase using isopropanol. After centrifugation, the RNA forms a pellet at the bottom of the tube, which is washed with ethanol to remove impurities, and then dissolved in water or buffer. One of the standout features of the tryzol method is its high yield. It can efficiently extract RNA from small amounts of tissue or cells, making it ideal for experiments where your sample size is limited. However, it's not all sunshine and rainbows. The tryzol method does have a few drawbacks. It involves the use of hazardous chemicals, so safety precautions are required. Plus, it's a longer process compared to other methods, which could be a disadvantage if time is a critical factor in your experiments. Furthermore, while the ability to extract both RNA and DNA can be an advantage, it can also be a double-edged sword. If you're only interested in RNA, the presence of DNA could lead to contamination unless additional steps are taken to remove it. So that's the tryzol method, but which one is the best? Let's compare. So, spin column or tryzol? Which one comes out on top? A question that might have crossed your mind more than once. Let's delve into this. The choice between the two methods often boils down to a few factors. First up, the yield. If you're after a higher yield, tryzol might be your go-to. It's known for its generous RNA output. Next, Consider the equipment you have on hand. The spin column method requires a centrifuge, while Trizol works with more basic lab tools. Lastly, the nature of your samples. Some samples, like those rich in proteins or fats, can be tough to work with. In such cases, Trizol can be a lifesaver owing to its proficiency in handling complex samples. However, it's worth noting that both methods have their rightful places in the world of RNA extraction. Each shines in its own light, catering to different project needs. The best method really depends on what you need for your specific project. But no matter which method you choose, verifying the purity of your samples is crucial. Now, how do we do that? Well, that's where NanoDrop comes in. This nifty little instrument measures the concentration and quality of DNA, RNA, and protein samples. When you're dealing with RNA, the results from the NanoDrop can tell you a lot about its purity. For instance, if your 260-280 ratio is around 2, it's a good sign. It means your sample is pure and you're good to go. But what about the next steps? You've got your RNA, it's pure. But now you need to choose between qPCR via cDNA and RT-PCR. Each method has its own merits. qPCR via cDNA is often preferred for its sensitivity and specificity, but RT-PCR can provide more detailed information about gene expression. So, whether you choose spin column or Trizol, remember to verify your results and choose the right PCR method for your project.